Thank you all for coming. It's a pleasure for me to be here. Traveled all the way over from MRB3. Got to get travel reimbursement?
Yeah.
Okay, awesome. I thought I'd give a little bit of a lesson in organizing symposiums. So I'd like everybody to say, not me. Okay, ready, set, go. Not me. Okay, the slowest people who say that end up organizing the symposium. So if you're in a room with Wendy Stone and Pat Levitt, and they say we want to do a symposium and you don't say it fast enough, you get to do the symposium. Just a word to the wise. So I am actually not an autism researcher, and it is something new and exciting that having worked at the Kennedy Center sort of got on my radar. And part of the reason that it got on my radar was from working with Mary Parton. And Mary had asked us to see a family who came down from Pittsburgh to be evaluated, to have their son evaluated, to see how he was doing with his therapies and to get a second opinion about the kinds of treatments that he was receiving. And so Greg and I ended up spending a lot of time with this family, and they were a really well-resourced family, a really well-connected family. And when we were talking to them, we just sort of were going through what it is that they're doing for their son. And one of the things that just absolutely blew me away was the amount of money that they were spending on special diets, on dietary supplements, on testing, for looking at things that were environmental toxins, that there were special dietary supplements, antioxidants that their son was on. And she had gone through about $7,000 worth of testing, where she sent in hair samples and blood and urine from her son. And there's really no data out there supporting a strong link between any sort of metabolic changes. And my lab focuses on understanding energetics. And so it seemed like a good opportunity for us to be able to provide a much needed bit of information to families who are really overtaxed to begin with and then stressed out with not having enough scientific information about what it is that could be increasing the risk of developing autism spectrum disorder. So I'm going to go ahead and get started. What I'm going to talk to you about today is moving beyond a classical understanding of cell biology and energetics. And we sort of all have this textbook knowledge about what it is in cells that makes energy and what it is that makes free radicals. And we all think, well, free radicals are bad, they're sort of nonspecific, and energetic failure is something that's sort of an irreparable part of the cell. Damage pathways. And so what I'd like to talk to you about is how those textbook understanding of how mitochondria, which are sort of the powerhouse of the cell, are really misunderstood and can be really important players in understanding very subtle perturbations, the kinds of perturbations that may underlie not just neurodegenerative diseases where we have strong linkage, but very subtle changes in cellular architecture. So I'm going to talk to you about how ATP and reactive oxygen species are unique and potent signals to change architecture, communication, and ultimately cell fate. And that there are cell-specific responses that are dictated by both intrinsic and extrinsic factors that can be influenced greatly by where a cell is in its developmental process. And we also are now able to use some very highly sensitive tools to assess the changes in energetic status. And some of that stuff that we're going to be talking about today is going to be a poster presentation by a fellow in my lab, Stephanie Hayden and Josh Brooks, who's also in my lab. So I will defer to them on that part of the talk. Okay. And we'll just hang out with this slide for a long time. Thanks, Greg. Oh, okay. So one of the biggest questions that we face as folks who work in neurodegeneration is, if you have a global change in energetics, how does that translate into a very specific cellular phenotype? And we know from genetic disorders that my lab works on in terms of Alzheimer's disease, Parkinson's disease, those kinds of things, that these genes are expressed throughout the CNS and even in other parts of the body, yet there's very discrete cell death that's associated with it. So what are the factors that dictate whether or not a cell is going to die specifically on a similar genetic background. And the model that we tend to use in the lab is stroke. And so we know that a global loss of energetic substrates through loss of blood flow doesn't cause all cells to die equally. In fact, regions like the cortex, the striatum, and the hippocampus are highly vulnerable to loss of ATP, whereas other regions, which have the same amount of blood flow loss, the cerebellum, are highly resistant to that. So how do we get to understanding what it is about individual cells? And a lot of what we need to do in terms of understanding individual cells is to get at understanding what it is about mitochondria that make each cell unique. And so this is a common theme when we're thinking about neurodegeneration, where these genes are commonly distributed. And so I'm going to talk about a little bit of that work that has helped us in understanding vulnerability of specific populations. So we know that the brain is highly dependent upon energetic reserves, and there's some nice sort of statistics that back that up, that while the brain receives 2%, it only comprises about 2% of body weight, it receives 20% of blood flow. And it also produces about 20 pounds of ATP in 24 hours. I'm just seeing if Pat's actually reading my slides. Okay. And it expends it expends 40 to 60% of ATP that's generated in maintaining ion homeostasis. So mitochondria are really the key intersection of understanding cell depolarization, energetics, and reactive oxygen species. And so let's talk a little bit more specifically about what it is about the mitochondria. The mitochondria, unlike neurons, or most neurons, continue to divide in postmitotic cells, and that this happens even more in neurons than other sort of supporting cells, sorry, Mickey, glia. And the half-life of a mitochondria in a neuronal culture is about 6 days. So they turn over pretty quickly, and most cells have several hundred mitochondria within them. So the range of numbers can go from 1 in a retinal ganglion cell to several hundred, but most of the energetic demand that happens during development occurs in humans when they're about 12 to 18 months of age, and in mice when they're about postnatal day 7 to postnatal day 30, and then that energetic demand dampens over time. So this is a time that we tend to think about as being a critical period in neurodevelopment as well, and some of these time points might ring a bell from talks that you heard earlier today. So about half of the proteins that are in mitochondria are tissue specific. I think that a lot of folks who are not cell biologists tend to think about mitochondria as being very homogeneous organelles, when in fact the complement of proteins that are expressed in a cortical cell mitochondria tend to be very different than the complement of proteins that are expressed in a cerebellar mitochondrial organelle. And the textbook knowledge of mitochondria belie the complexity of the organelle. And what I'd like to do is to touch on a couple aspects of that. So this is sort of your traditional Stryer model of how mitochondria work. So you have glucose that comes in, goes through the Krebs cycle, and then provides reducing equivalents to fuel oxidative phosphorylation. And when we look at oxidative phosphorylation, these are the complexes that we look at. And when we think about what the mitochondria look like, waking my computer up. Lunch was good, but wake up. You can do it. Okay. The knowledge that these organelles generate about 90% of cellular ATP and 90% of free radicals is true, and they also are the gatekeepers of cell death, meaning apoptotic and necrotic signals. And so this is a picture where you see the nucleus is stained blue with DAPI, the mitochondria are stained red with a mitotracker dye, and the green is a marker called LC3, which helps to autophagocyze injured mitochondria. So these are some of the kinds of things that we do in the lab. And so again, looking at what we think about as a textbook mitochondria, you see this outer membrane, which is somewhat rigid, an inner membrane, which contains the complexes that are associated with oxidative phosphorylation. And we tend to think about them in terms of having a relatively consistent stoichiometry. You have one complex 1, one complex 2 that feed electrons through to complex 3 and complex 4. Well, that's completely untrue, and it varies dramatically depending upon what organ you're in. And so a more representative image of what a mitochondria looks like is actually this, where this is a reconstruction that was done of some EM work, where you can see that this idea of the inner cristae, which is in green, is really much different than sort of our textbook understanding, and that the stoichiometry that I'm not going to have a chance to talk to you about today tends to be very, very different in any given organelle within a neuron in particular. So when we are thinking about the kinds of signals that mitochondria are integrating, really we're thinking most on the top of our head about energetic status. And we know that when we're looking at markers of how cells are stressed, we can look at ATP, and that the generation of ATP and the production of lactic acid are intimately linked, and that what we can do in cell cultures, in brain imaging experiments, is to start to link the loss of ATP with the production of lactate. So this is a stroked brain, where you can see on the left, you're right, your left, sorry, somebody else did that earlier. Your left, the regions where the pH is starting to drop down and where ATP is beginning to be compromised. So ATP is starting to drop out in the blue regions, and the pH starts to sort of cack out and become highly red in these pseudo-colored images. And changes in substrate delivery or glucose delivery tend to elicit these changes in ATP production and lactate generation in the order of minutes. And so these are things that can be helpful for us in terms of thinking about markers of stress, so that if we move away, this would be easier with a pointer, but it would have to be a monster pointer far, far away. Here, I'll do it this way. Can you guys see that? Yeah, okay. So that if we move away, so if we have glucose coming through in glycolysis, and if our oxidative phosphorylation complexes start to go offline, what happens is that pyruvate then gets shuttled out through lactate. And so in individuals that are experiencing energetic stress, you can look peripherally in blood and urine and those kinds of things to see an increase in lactate generation. And this is a pretty common clinical tool that we use. It doesn't tell us exactly where the deficiency is in the mitochondrial complexes, but it does begin to say glucose utilization is subprime, because once we move away from aerobic respiration and the ability to produce ATP through oxidative phosphorylation, what happens is the amount of ATP that we're producing decreases dramatically. We go from producing 34 molecules of ATP down to 2. So not only are these potent markers, but the changes in ATP and the resulting changes in pH and in reactive oxygen species that are generated are also potent bioactive molecules. So when we think about ATP, we tend to say, well, we need that in order to keep our cells happy and our membrane potential at its optimal level. But it's actually quite more complex than that. And so I want to show you work that's not actually from my lab, but demonstrates this well. And that's looking at microglia. So microglia comprise about 10% of the cells in the CNS, and they have highly complex roles in maintaining appropriate neural communication and working to ensure that cells which are damaged are removed. And so they also have been linked to autism spectrum disorders. And this is an image that Carlos Parda had shared with me, where he's looking for activated microglia. We're where he's looking at activated microglia. So the panel on the right is one in which it's the cerebellum section from a postmortem brain of an individual who had autism. And you can see that the cells that are stained brown are actually having markers of activated microglia. So how can we then go and link activated microglia to traditional signs of energetic dysfunction? And there was a wonderful study that came out 2 years ago in Nature Neuroscience that did that. So let me see if this movie will work. And what they did in this work, this is some beautiful 2-photon microscopy work that was done in which they took the skull off a rat, and the rat has microglia that are GFP labeled. And all they do in this animal is, you can see this sort of red area. This is a micropipette which is going into the cortex. And all they're doing in this is just dumping a physiological level of ATP, which you would normally see intracellularly. And you can see, this is a movie that lasts about, is taken over a course of 60 minutes. When I say you can see now. So they dump it on, and you can see those microglia with only the signal of ATP just immediately go to the site of injury. So this is a very potent signal to get microglia to the site of injury. And it doesn't require anything other than the energetic substrate which we use in mitochondria. So I think that when we're talking about what it is that mitochondria are doing, it's really more than they're producing ATP and maintaining ion homeostasis and those kinds of things, but that when things go awry and cells start to lyse and ATP comes out, that is a really key molecule that will get other cells to the site of injury. And microglia aren't just going in and encapsulating injured cells, but they also change architecture in a pretty dramatic way as well. There they go, pretty. So I had a backup slide in case my movie didn't work, so we'll just go to this. So in terms of thinking about regional specificity and what it is that makes cells vulnerable, there's been some work that Tim Greenemeyer, who is a very well-known neurodegeneration researcher, has done, looking at pesticide use and the vulnerability of workers to developing Parkinson's disease subsequently. And so he did animal model work in rats and in baboons where he exposed animals to a complex 1 inhibitor called rotenone. And so what you can see in this, these sections from rats, the top is just complex 1, they're stained black because they have a lot of complex 1, which is part of the oxidative phosphorylation chain. And on the bottom are sections that were taken from, this is complex 1, complex 2, and complex 4. On the bottom is an animal that was treated with rotenone, and you can see that there's very little selectivity in terms of the depletion of complex 1 that occurs. And complex 2 and complex 4 remain intact. So they look at these animals about a week after they get the injury. And then they go in and they look to see, well, what are the cells that are dying? And if you go with a standard prediction, you would say, well, complex 1 is gone, your ability to make ATP is gone, well, all of your cells should be gone. And that actually is not the case. There's very discrete regions of injury. And that's been shown in the next series of panels here, where they look to see the staining of tyrosine hydroxylase positive cells and their projections into the striatum. You can see that those are very significantly depleted in animals that got rotenone treatment. So he went on to look at what are the consequences of those kinds of, Okay. He went on to go and look at what are the consequences of those kinds of treatment and look to see that there were aggregates that were being formed as proteins are being misfolded. And so this tells us a lot about how global changes in energetic status are getting translated down to very specific cell changes. And when we're thinking about if you're having a loss of substrate, really the substrate we're depending upon most is glucose. But if you add more glucose, that is actually not a reasonable therapy, and we know that from stroke literature and stroke clinical work pretty dramatically, so that if you increase glucose in a patient that's having a stroke, where they are deficient in glucose because they've lost blood flow to the brain, it's actually highly deleterious, and it tracks really well with having a very, very poor outcome. So that the more glucose you put in, when your mitochondria are unable to process it by making the lot of ATP that they need because they don't have oxygen on board, what happens is that people start to go into lactic acidosis. They produce a lot more reactive oxygen species. The blood-brain barrier starts to break down. That there's edema, which actually turns out to be one of the biggest pathological problems that's associated with this. And that what eventually happens is that individuals start to undergo hemorrhagic transformation, where the vessels in the brain start to explode. So simply adding substrate in is really not going to be something that's particularly helpful. So when we're thinking about how it is that the mitochondria fail, and we're thinking more about what are the environmental toxins that can be predisposing mitochondria to failure, there are a couple ways that mitochondria can fail. And one is through direct direct inhibition of the electron transport chain, and that's things like rotenone, which act as complex 1 inhibitors, malonate, 3-nitropropionic acid, those kinds of things. There are also uncoupling agents, which start to poke holes in the inner membrane of the mitochondria, so that the proton gradient that mitochondria need to generate ATP is dissipated, so that the mitochondria essentially then go offline. And the respiratory chain can be altered acutely or chronically by drugs that are on the market. And this is something that's a very rapidly growing field and something of keen interest to a number of drug companies, because when drug companies screen drugs right now, they tend to sort of look at things like, is this a drug that will cause an arrhythmia? Will it be activating altering potassium channel currents? Is there an abnormal immune response that's associated with that drug? Is this a drug that will cause hepatotoxicity? And really, of the drugs that have gone through and gotten black box warnings, it turns out that a lot of them have mitochondrial liability, meaning that they are acting at the electron transport chain or other parts of the mitochondria to impair mitochondrial function. So Pfizer took it upon themselves in the last couple years to get much more aggressive in screening drugs that are on the market and moving through the clinical trial pipeline. And they found out that about 50% of the drugs that they're moving through pipeline or are available through clinics now have mitochondrial liability. So in terms of the drug discovery effort, this tells us a lot more about what is a profile of an agent which is going to be potentially toxic. And so of the agents, I know you yellow lighted me. Of the agents that are on the market, the ones that have been removed, all of the ones that have arrows beside them are actually quite potent mitochondrial toxins. So in terms of thinking about the major nutrient sources that are coming through in glucose, we've talked about these issues of how just adding glucose isn't sufficient, and that there are other features that are associated with the kinds of mitochondrial liability, which are things that we need to, that actually have some hallmarks of the developmental disabilities that we're looking at that might suggest to us that mitochondria are not well functioning. And those are sort of listed here. I'm not going to run through them all, but certainly these ideas of lactic acidosis, changes in mitochondria structure and function are very important, and things that, other than lactic acidosis, are really things that we have not looked at well in individuals with developmental disabilities. So the mitochondria is a very complex organ which can fail in a number of very important ways. And one of the ways that I want to talk to you about in my last 2 minutes, Dr. Levitt, is production of reactive oxygen species. So reactive oxygen species can be produced by a number of different subcellular sites. But really the predominant one in terms of producing about 90% of reactive oxygen species is the mitochondria. And so unchecked reactive oxygen species, or free radicals as we call them, have been implicated in virtually every neurodegenerative disease, as well as some of the chronic neurodevelopmental diseases as well. So what are the measures that are out there? Some of the measures that we'll talk about in Josh and Stephanie's work will focus on some work that came out of BOSS study. So what happened is NIH sort of got sick of all the investigators saying, well, there's a ton of different ways that we can look at free radicals that are being generated. You can look at DNA damage, protein damage, lipid damage. So we're going to put you all in a group where we go head to head with the amount of free radicals that are produced and come out with the best marker. And the best marker was something that we do at Vanderbilt. Jason Morrow and Jack Roberts have really perfected this technique in looking at lipid peroxidation. So this was a study called the Biomarkers of Oxidative Stress study, and the isoprostanes which are formed from that is something that you can learn about a little bit more this afternoon. So in my last little bit, I'm going to just tell you that reactive oxygen species like ATP are not just random destructive agents. They have very discrete signal transduction pathways, and that if we look at isoprostanes and different categories of isoprostanes, they activate discrete signal transduction pathways, and some of those things are molecules like P66-SHIC. So P66-SHIC is an adapter molecule, and it turns out that if you have a serine 36 residue and you cause a mutation in it, you can increase the lifespan of an animal by over 30%. And that what happens is P66-SHIC moves to the mitochondria and alters the coupling of the electron transport chain. So there are a number of tools that we can use to understand signal transduction, energetic status, and that we can look at the factors which influence energetic demand during development, that there are mechanisms that are available to compensate for that, both through drugs and endogenous mechanisms that we can capitalize on, and being able to provide some therapeutic relief if there is actually a deficiency in electron transport chain activity. Our lab is very interested in taking fibroblasts from individuals with autism and being able to look at isoprostanes and energetic coupling. So I'm going to go ahead and stop there. Thank you.